Good morning. Thanks, Ron, for the introduction. Thanks, Monzi, Allison, for the invitation. It's always nice to be here to speak with ILSI. To start out, I'm going to go through a little bit of overview of what I'm going to talk about here. I'd like to first talk about a little bit about the food contact notification process, so what we're looking for in safety assessment and in food contact notification, so then we can get an idea what signs we're looking at and where we might find some holes, where we need to move forward, some new challenges. And so the basic overview will be, we'll start out with some toxicological aspects of safety assessment for food packaging materials, and then on to some more detailed information on the FCN review process, our scientific recommendations, and recently some common deficiencies we've been seeing. So there's some areas we can work on. And then I'd like to talk about some new guidance we have hopefully coming out in the next few months, and this is where we've put together a lot of the newest science and hopefully where we can help advance our safety assessments in a specific area. And then a little bit onto nanotechnology and other challenges, all good areas I think we can work on together. So first, I should define what food contact substance is. It's what we look for in food contact notification, and it's very important here. It's any substance that's intended for use as a component of materials used in manufacturing, packaging, transporting, or holding food if the use is not intended to have a technical effect in the food. I think that last component there is very important. It's not intended to have any technical effect in the food. I know some of the pre-meetings we've had have heard about smart packaging, active packaging. These are where you'd have materials that are intended to have an effect on the food, increased shelf life, effects like this. That would not fall under our regular food contact notification, food contact substance. That would be more along the lines of a direct additive, and we'll go through our division and petition review. A food contact notification is somewhat unique too in the regulatory system. Now, since we have a 120-day mandated deadline after we receive a complete submission, that's once we determine that submission is complete. If we don't object in 120 days, it can go to market. Any notification that does become effective is listed online, so you can see who owns that notification and what the notification is for. And that's the next point is along that line too, is that a food contact notification is exclusive for that manufacturer or supplier. So if someone else wants to come in and use the same material, they got to submit their own food contact notification. And the major purpose for a food contact notification is to demonstrate human safety. I should say, you're not just efficacy, but you do have to demonstrate efficacy in that you have to demonstrate the minimum amount needed to achieve the desired technical effect. But the overall goal is a safety assessment, make sure the product is safe before it goes to market. And what do we mean by safe? We do have a legal standard here. This is our standard safety, the reasonable certainty of no harm. And I like this quote from a previous congressional report. It gives a very good overview of what we mean by safe. And that's the concept of safety used in this legislation involves a question of whether a substance is hazardous to the health of man or animal. Safety requires proof of a reasonable certainty of no harm, and we do that to our best of our abilities. Will result from the proposed use of an additive. It does not and cannot require proof beyond any possible doubt that no harm will result under any conceivable circumstances. I believe that last sentence puts things in context there. I mean, we could test forever until we find something, but that's not reasonable. Although there are a number of groups starting to push those ways, and even to the point of just saying let's use precautionary principle instead because there's something there, although we haven't tested for it yet. But that's not the way we work. We look at safety assessments, risk assessments, make sure we have a full package here. When we look at food contact notification from the toxicology side, we're looking at the investigation and safety of the food contact substance and any constituents, any impurities, any breakdown products, anything that's going to be part of the food contact substance that's going to migrate to the food. And in a food contact notification, we lay out the toxicology as a safety narrative. This is an area just to give I compare this to an executive summary. It needs to tell a good story here. Describe the scientific basis of the safety determination. And then in a comprehensive toxicology profile, this is where you need to put all the unpublished, published safety studies, all relevant information, and lay out the full safety assessment for our evaluation. So what type of science do we normally recommend? We only ask for toxicology data that we need to establish a safe level of consumer exposure to food contact substance and its constituents. We do use an exposure-driven tiered approach, and I'll show the recommendations on the next slide. And when we have impurities or constituents that are carcinogenic, we do require a risk assessment for those. I said, so here's just the basis of safety evaluation for food additives. This goes for food contact as well as direct additives. Our safety standard is the same, and we look for similar elements in a safety assessment. One is the structure and physicochemical properties. We need to know what we're looking at here, and then a dietary exposure to these compounds, and toxicity data on the compound itself, or occasionally we can use structural analogs as supporting information on safety. And when we do look at the toxicity, we look at two different areas, neoplastic, non-neoplastic endpoints. So for the neoplastic, carcinogenic endpoints, types of studies we're often looking for are chronic bioassays, 2-year assays, rodents usually, or depends on the exposure. It could be genotoxicity batteries to give us more of a hazard identification for potential carcinogenicity. And then on the other side, we look at non-neoplastic endpoints, primarily systemic toxicity, often subchronic studies or even longer-term studies. And when we look at that data, if there is any markers or additional information that suggests we need to look further, either repro, developmental, neuro, somewhere else, we recommend those type of studies as well. So you can find this information online on our guidance. I put the link down here if you like. We do, like I said, we have a tiered approach here. As the exposure gets higher, we ask for more information. I guess I can use a pointer here. So we start out just at the lowest exposure. We look for what carcinogenicity information is available in the literature. And as the exposure increases, we ask for a little bit more genotoxicity studies, and we go to systemic toxicity. And once we get up to about 1 part per million, this is getting into the area where it's almost considered a food additive. We may look at some different studies for that. And what you see here, carcinogenicity, we still feel is the most sensitive endpoint, and we still do, like it says, this is our minimum testing recommendations. We still do believe that the dose makes the poison, and that's the principles we follow unless the data suggests otherwise. And in recommending studies, we do have what we call our Redbook 2000, the Toxicological Principles for Safety Assessment of Food Ingredients, and you're welcome to go look at this as well. This has a lot of good advice, a lot of good guidance on types of studies to conduct, proper conduct of the studies, GLP, lots of information there. And you can tell Redbook 2000 stands for the year 2000, so it has been quite a while since it's been updated, but we're taking an initiative this year to really work on updating that. There has been a lot of science available. It's one of the reasons we're having this meeting today is because the science continues to advance, and we'd like to update our guidance to incorporate a lot of that new science. So I did include a link. I think there are contact information there if you'd like to talk to somebody about updating the Red Book. But the goal is to expand the scope of the Red Book to emphasize the principles of safety and risk assessment that are shared across different regulatory contexts for foods and cosmetics. So like I said, what's important is that in Division of Food Contact Notifications, we've been seeing an increase in the number of submissions over the last few years, which I see as a good sign. That means industry is producing, innovating, more information is coming in. You're working us to death, but that's okay as well. But recently we've been seeing a lot more common deficiencies in the safety assessments. I just want to point out a few here. So if you're writing up a food contact notification for new material, keep these in mind. One is incomplete or disorganized safety narrative or comprehensive toxicology profile. Like I said, we need to see a good story, write out a full safety assessment. The next two kind of go together, insufficient toxicology data or inadequate assessment. Often studies might be cited and might not be submitted, so we can't review them for our own evaluation, or they aren't reviewed correctly. And in cases for risk assessments for carcinogenic constituents, sometimes when there is only hazard data available for a substance, often the analog can be used to, in its place to do the risk assessment. But often what we've been seeing, the bridging data saying why this is the best analog for the constituent is often missing. So on to more advances in the science. So this is an area where I think the science has advanced enough in the last decade, 2 decades, that we're now able to pull this information together and start issuing a new guidance, which we'd like to see come out in the next few months. on specific subpopulation. Now, normally our food contact guidance captured the whole general population. Now we're have enough information, enough science to support breaking out one specific subpopulation. That's for food packaging for infants. It's always difficult to say. If you say infant packaging, it's not new packaging, it's packaging for infants. So this was tricky writing some of the guidance on this. Kirk always likes to say about infant consumption. No, we're not eating infants. It's consumption by infants of materials. So I just want to go through some of the ideas and some of the things that we're proposing in this new guidance and the way we can look at this special subpopulation, which we're looking at the 0 to 6 month period. And this is, as everybody would know, this is a very critical period of development. There's a lot of turnover cells, organ development, immaturity in different systems. So we felt, and there's enough developmental data now available to look at this postnatal period separately and really do a more precise safety assessment for the specific population. And one of the things we look at first, like I said, exposure, so infant dietary intake. We'll use some NHANES data. And our traditional values, we use 300 grams of food per person per day, usually. However, when we look at this NHANES data, we were able to get a better calculation for an infant 0 to 6 months of 6.3 kilograms in body weight, 140 grams kilogram body weight per day for food. So these are now the numbers we're using to calculate exposures. And like it says here, these values were indicative of the highest food intake to body weight ratio to represent the worst case exposure scenario. So, minimum testing recommendations. So I showed this slide earlier. However, another change we made here, it's not a real change, but our guidance was, you could say, somewhat simplified, make it easier to read, where we used to do micrograms per person per day, and that was based on 60 kilogram body weight for a person. However, if we remove that body weight, that factor, we can now normalize this out to micrograms per kilogram body weight per day. And it's the same tiers, same exposures across here, so no change in what we're recommending any tier. It's just putting this into different terms so we can compare different populations, and it also helps the toxicologists because anytime you have a tox study, dosing is often micrograms per kilogram body weight per day, so it's much easier to compare straight across. And if we move forward later on in the future and we find that there's specific products for a specific population, we can move things around and everything will come right back to the same tiers. So what are we looking for in a safety assessment for infant products? Like I said, for baby bottles, packaging for infant formula. Taking a slightly different approach here. We don't have specific studies lined up right now. This is more of a case by case. You can think of it as personalized medicine for a food contact notification. We look at each one separately. And these are the types of questions we think about, we like to ask other people to think about, because the science has advanced significantly, but it is still changing, it's still developing, and we still have areas to work on. We don't want to pigeonhole ourselves in one area where if one study works better in a certain case than another one, then we want to go with that. We always want to incorporate the best science, and this type of guidance will be able to advance with advances in science. And some of the questions we think about. So do the available toxicity or chemical structures suggest a concern for developmental toxicity? Is there a reason we need to look at additional testing? You know, in certain cases, the infant may be more sensitive and may be less sensitive than an adult. So can we focus in on where those differences might be and what we need to look at? Next question is, would any endpoints identifying juvenile or animal toxicity study be expected to result in a different effect or change in magnitude or sensitivity of an effect in neonate or pre-weaned animal? And for this one, let's give an example. So for, suppose we had a subchronic study, which we normally recommend, and we found that there was liver toxicity was the main effect in that study. Or not, I'm sorry, not liver, but kidney toxicity. And we know the kidney is not fully developed till 1 year of age or older, so that could give us a marker of now if there's a product for infant use now, maybe we need to go and look at a specific study for liver toxicity during the developmental period so we can have a more focused approach on our testing. And also using information we already have to help guide us. Other considerations and things I'll say later, but I think we need to include more often. What other type of data may be useful to inform age or species-dependent differences? For example, absorption, distribution, metabolism, excretion studies, mechanism of action, pharmacokinetics, toxicokinetics. I think this information is very useful for really using the toxicity data we have available in a different way. Instead of having to do a lot of the other developmental neurotoxicity testing studies or two-generation studies, or I think we can use the science in a different way to help get to the same answer. Did the available data testing address the concern identified? Can the available information adjust the variability or differences between infants and adults? And for example, I have another example we saw not too long ago. This was a case where we had adult rodent data, and what's unusual, we did have adult human data in this, and we had developmental data in rodents as well. And through PK analysis and looking at this additional information, we were able to tell that infants were likely to be about 20 times more sensitive to this compound than were adults, which was higher than our normal intraspecies variability safety factor of 10. So in that case, we did raise the safety factor for this product. And some potential use of PK data. I'm a very big fan of PK data. I think it resolves a lot of issues or resolves a lot of questions and uncertainty around specific topics. And some areas of uncertainty that could be resolved or could be very useful to test using PK data. The dose delivered to pups in most developmental toxicology studies. We never know what the dose in the pup is unless we test the pup blood itself. But most studies don't do that. Usually a pup is assumed to receive a dose through lactation from the mother. But if we don't have the PK data or the ADME data to know that the material is going into the milk or it's going into the milk unmetabolized, we really don't know what's getting into the pup. So PK data can resolve a lot of this uncertainty of what is the pup exposure if we're looking at a true postnatal developmental study. Differences in pharmacokinetic profiles between adults and juveniles or infants as well. Like I just mentioned, we can, and I'll give another example on the next slide here, the differences and how we can apply those. Differences in pharmacokinetic profiles between humans and laboratory animals. And I think we'll see a little bit of that in the next slides too. There could be a different sensitivity, differences in metabolism, and issues that can help us refine how we extrapolate from rodent studies to humans. And like it says there, PK data may help refine these areas of uncertainty, possibly modify safety or uncertainty factors used in the safety assessment. And here's one example. I pulled this slide from a previous talk I had, so you don't have to look at the words on the side there as much. But what was important here, this was a PK study done measuring internal dose of a compound in mouse, rat, and monkeys. And what we saw that's an adult, all three species, metabolism completely eliminates the compound. And juvenile, same there. However, as we get younger and younger, it shows that the metabolic capabilities in the mouse and the rat were much more immature than the monkey at the newborn stage. So in fact, the monkeys, or not the monkeys, the mice and the rats were getting much, much higher internal dose. Although they were given the same dose as a monkey, their internal tissues, internal serum levels are much, much higher. And this was really important for one of our assessments because most of the data in neonatal rodents showed a very high range of toxicity. But if you just took the toxicity data and compared that across to primates, it'd throw you off. You expect this type of toxicity in the primates. But in fact, when you see that the dose disappears, is nearly completely metabolized in primates, there's much less concern there. So it gives us a very good comparison. And we can use it quantitatively as well. And then taking that data, and here's another slide I've pulled, which I think another fantastic study here by Fisher et al., using physiologically based pharmacokinetic model. So really using the computational aspect from that pharmacokinetic data to connect the dose-response relationship to that internal dose. And these are great tools for extrapolation from route to routes, between species, between ages, between doses. However, as with all models, they're only as good as the data that's gone into them. But I think this is an area we can always work together and still has room for advancement and definitely application in the regulatory setting. And just to give you one more example, the same group, Fisher, who works at FDA now, but also collaborates with EPA, and EPA has recently released a call for their review of their draft assessment of, or not draft assessment, but they have a draft biologically based dose response model, which is essentially a PBPK model, although they're taking it one step further and using this type of model to try to predict the toxicity endpoint based on the exposures. And one last piece for the infant safety assessment, and that's for the constituents and impurities, if there are ones that are carcinogenic. We've modified our lifetime cancer risk formula, and this is kind of based on EPA's evaluation and the same way they do that. So in this case, we break it apart and add an age-dependent adjustment factor, and EPA has done a very nice analysis of this, of the higher turnover cells, potential for increased carcinogenicity in these during developmental period, especially between 0 and 2 years of age. So for our risk assessment, we take 0 to 6 months. I've lost the marker up there, but in the first one, 0 to 6 months, we have our unit cancer risk times really a safety factor, an age-dependent adjustment factor of 10, times our infant exposure times the percentage of the lifespan. And then same for 6 months or 2 years, and 2 years through 78 years, and we add that together to get our total lifetime cancer risk. So that takes into account increased sensitivity during that early developmental stage. However, if there's more information, PK, other types of information, safety factors can always be modified with supporting information. Some additional projects related to our new guidance and our safety assessment for infant products. We did publish a paper on the state of the science and considerations in evaluation of safety. If you'd like to look at that and see how we've laid out many of developmental toxicity testing studies, paradigms, pros and cons of each. We also have a couple projects going on currently. One is examining 1 and 2 generational developmental and reproductive toxicity studies in our databases, determine which ones may be the most informative for safety assessment. And then we have another project in collaboration with our colleagues at the Center for Drugs and Evaluation, for Drug Evaluation Research, and that's investigating potential utility of juvenile animal studies protocol for relevance to our safety assessment. I know one of the things that came up in our pre-meetings was discussion of nanotechnology, and that seems to be a popular issue right now. Some of the important points to talk about nanotechnology. We do have some agency-level guidance, really talks about how you evaluate the structure, the identity, exposure for nanomaterial. We've had a few submissions for food contact notifications. However, usually once we get into the evaluation, it turns out they're not really in the nano size, but so it doesn't change things that much. And then at the bottom, is it safe for the intended use? Is there unique toxicology? And well, that's really the question right now. This is an area we need to do additional work on. What type of is there unique toxicology? What types of studies do we need for the toxicology? Do we need to modify our current studies based on the properties of the material being tested? We have a few studies going on at our research labs in Arkansas. Some of those are just now starting to finish up and be published. We hope to start incorporating that information as well. So I see that as an area we can all work on together. Some other areas, challenges with new science interpretive tools. I wouldn't say these are new science anymore. They've been going on for a while, but because data collection takes so long and we learn a little bit each time we do another study, I wouldn't call it new science, but I think the way we use it now, we're getting to a point there's enough data available. We need to start combining this data, start looking at it a little differently, and how we apply it to regulatory safety assessments, start looking at validating some of these types of studies or combinations of these studies. For example, high throughput screens in ToxCast, in vitro assays, computational toxicology. There has been a big push on increased focus on mechanism, mode of action, adverse outcome pathways, thresholds of toxicological concerns. It's one of my favorite just personal topics. And making better use of existing data from our series database, quantitative structure, activity relationship, read across. And like I was saying, one of the things I think a challenge, something for the future that we can really work on, maybe the last point here is, I know separately people like to take either ToxCast, high throughput screens, they're looking for the right in vitro assay that will predict some type of in vivo toxicology. I'd like to see a project where we turn it around the other way. Take all the vivo data we have and try to do an extrapolation to what type of combination of these high throughput screens, in vitro assays that will be predictive for those certain areas of toxicity. I'd like to see that type of validation come out where we can see combinations or patterns of studies that we use to get to that point of being able to quantitatively do a safety assessment or a risk assessment and push us towards the future and maybe get to the point where we don't need as many animal studies anymore. But I think that's going to really take a lot of our bioinformatics and mathematicians to really work out these patterned information. And that was my last slide. Thank you.
Hey, Jason, we do have time. We do have time for a couple of questions. If anybody would like to take this time to ask Jason a question.
Great talk. Thanks for all the information. So I'm from an infant formula company, and I'm going to ask an infant formula question, but I think it's probably broader than just that. So I think what we're seeing is that there's some really good stuff coming out on the hazard side where more sophisticated models are coming out, like the benchmark dose, that really take advantage of all the data that's available. So on the exposure side, and I know we've got Keenan coming to talk a little bit later about exposure, is there an opportunity with these FCNs to make arguments around maybe not understanding that the worst case estimate of exposure, it plays an important role to triage, but is there an opportunity to discuss what true exposure would be and being able to use distribution of both consumption and occurrence to refine that exposure assessment as part of this FCN process?
Yeah, I believe that's the case. I mean, more realistic, I think more precise we can get, the better data we have. Although, I think you should save that question for the next talk. Is that right? Okay. Yeah, Kirk is our chemist and our exposure expert, and he'll talk a little bit about that. Yeah.
Thanks.
Anyone else? Greg.
Jason, in terms of those of us that are doing this user compliance and migration testing for our package components. For those of us in the supply chain that are doing the migration testing of the packaging, user compliance testing, so their packaging has already been, the food contact substances have been approved, and they're using these packages on a daily basis, and we're monitoring it. Should we be looking at not only, should we add migration testing related to the exposure scenario for infants also? And is that what's going to come out in the guidance? Will there also be the migration testing chemistry recommendations similar to the one for the normal guidance?
Yes, there will be a chemistry component of that to look at specifically the products for infant formula and breast milk use.
Thank you, Jason.